Yeah, in the early days of sequencing, the real innovation around next generation sequencing, or the sequencing that propelled us into this new era of sequencing, was short read sequencing. And that was basically 100 to 150 bases of sequence. So you took your genome, you chopped it into little bits, and then you would take those small fragments that basically gave you a small window into the genome. You would match them to the reference genome. You had to have a reference genome to
Hmm.
match them. And then you could say, okay, what are the base pair differences in this region? So it was very limited, and it was really specific to what we call single nucleotide polymorphisms,
Mm.
or specific base changes. So there's four letters to the DNA alphabet: A, T, C's, and G's. And basically, when you have a slight change in that alphabet, you actually could cause a downstream change in the plant. So in those early days, yeah, we would just take the short reads, we'd map them back to a reference genome, and then we would see what the differences are. Now, imagine the case where that reference genome does not include a very important gene. So then you sequence, you make this short read, you try to map it back to your reference genome. There's no place to put it, no place to map. So, in other words, those short reads are blind to that region of the genome. So, fast forward to our study where we actually generated all these genomes using long reads. And so, long read sequencing generally is characterized by reads that are 10 kilobases plus. So, two of the technologies that we use in my lab are PacBio, or Pacific Biosciences, and then Oxford Nanopore Technologies, both of which make reads that are on the order of 15 kilobases to over a megabase of sequence. So now you're looking at much larger stretches of sequence, so you could map them back to your reference.
Mm.
However, with these long reads, what we're able to do is actually assemble the genomes into new reference genomes. And now this is where we actually like to change the language a little bit. We don't think of references anymore. We think of the pangenome as that actually the reference now. So this is the reference pangenome, and each individual genome inside of the pangenome is what we call an anchor genome. And we can now look at the variation in each individual genome that might have been missed if you were just using the reference genome.
Interesting. Okay.